Hi, I'm Mackenzie Cundiff, and the gene I will be discussing for my Bio 19 honors presentation is the HTT gene and its contribution to the Huntington's disease. So before we get started, I wanted to start with the table of contents to kind of let you know what we'll be talking about. First, the introduction, kind of just the normal function of the gene. Second will be the gene research, so answers about the actual HTT gene. And then protein research, so the product of the HTT gene due to the mutation. And lastly, the conclusion, just kind of the wrap up where current research is at and what therapies look like. So first, just the introduction of HTT and Huntington's disease. I have this little image showing the early symptoms of Huntington's disease. So there's moodiness, fidgeting, cognitive issues, personality shifts, depression, trouble focusing, and muscle twitching. So definitions, the HD is a rare inherited disease that causes the progressive breakdown or degeneration of nerve cells in the brain. This normally results in movement, cognitive, and psychiatric disorders. The HTT gene is a gene that provides instructions for creating the Huntington protein. Every human has the HTT gene. It is known to play a huge part in the neurons in our brain as well as normal development, although it is most widely known for its abnormal glutamine stretch in the N-terminal sequence, which leads to Huntington's disease. So this is just kind of like a summary of what I've said. The Huntington's disease is an autosomal dominant neurodegenerative disorder. It is caused by the CAG trinucleotide expansion in the HTT gene. This encodes for the abnormality within the Huntington protein. So, some facts is that 30,000 people in the US actually suffer from HD. 200,000 people in the US are actually at risk for developing it. The age range for symptoms that commonly start is 30 to 50 years old. So, this kind of shows it broke down in the molecular level. This shows at the very top the HTT protein and the exons. And then it shows the wild type, how it has 9 CAG repeats, and then how the mutant has 45 CAG repeats, and then that's a different mutant that has 36. As you can see from the models below, you can really tell a difference in those due to the excessive amount of CAG or the glutamine repeats. So moving along straight into the gene research of the HTT gene. So this is just kind of another image. There's quite a few in this presentation, but I just think it helped me understand it most, seeing it in different perspectives. So the top is just the normal healthy neuron that is produced from less than 35 CAG repetitions. So you have that normal HTT gene and then the healthy neuron in the healthy brain. Then you will have the mutated HTT example, which is greater than 35 CAG repetitions, which results in the expanded polyQ region, which is the mutated HTT protein, and then the neuron degeneration in the pathological brain. So basic gene info is that the chromosomal location is at chromosome 4, the gene size is 180 kilobytes, and introns are 66. The average intron length is 2,360 base pairs, and for exons, there's 67. They range from 48 to 341 base pairs, an average of 138 base pairs. I know that's kind of a large range, but that's on average. So to continue with that, it is expressed throughout the body, mostly expressed in the brain and the testes. The subcellular location of gene is dynamic and very complex. How is it regulated? The HTT promoter has high GC content and lacks the TATA and CCAAT regulatory elements. Multiple regulatory regions can be found within the highly conserved regions, but results are not consistent on what truly regulates the gene. Some transcription factors is the polyQ or the polyglutamine tract is an expansion that increases the HTT and DNA interactions with the binding to recognition elements of factors that function altered HD. HTT is regulated in the transcription level. So, HTT is a largely ahelical and has 3 major domains. The amino and carboxyl terminal domains have multiple heat repeats. So, heat stands for Huntington, elongation factor 3, protein phosphatase 2A, lipid kinase TOR. So we can go more in depth for what each of those mean. So Huntington is the highest level of activity in the brain, involved in chemical signaling, transporting materials, and binding to other proteins and structures. The elongation factor 3 stimulates the binding of amino acid tRNA to ribosomal A site by assisting in the release of deacylated tRNA E site. So that's the exit site. The protein phosphatase 2A is a multi subunit CAB, very common in neurodegenerative diseases such as HD. CAB, that stands for the scaffolding, regulatory, and subunits. So moving on to the lipid kinase TOR, it's the central hub for the cell growth and cell proliferation. So moving along to the protein research, this is another image. Like I said, this has kind of been previously mentioned, but just another view on it. This is a healthy gene, so less than 35 repeats of the CAG sequence. Um, and that's what that looks like. And then you can see the Huntington's disease gene, which is greater than 35 repeats of the CAG or the glutamine. And you can see that expansion of the polyQ. Like I said, another image, they're all a little bit different and help me with my understanding. So hopefully they'll do the same for you. So this is more like the helix of the non-mutated Huntington gene. And this is the non-mutated protein that is in response to that. And then the expanded polyQ what would be the mutated Huntington gene. So basic protein info. The size is 350 kDa, which is pretty large. Different forms, there's 22 different splicing variants due to the alternative splicing. The position is on the short arm of chromosome 4 at position 16.3. Its half-life is 24 hours and is degraded by the proteasome and autophagy. The domains, there's two interacting regions. One is the HAP1, which regulates vesicular transport by interacting with motor proteins and signaling molecules. The dynein is what Huntington interacts directly with to facilitate vesicles motility along microtubules. So moving on to the amino acids, there's 3,144 amino acids. Most are in which what is previously stated, the cytosine, adenine, and guanine CAG sequences coding for the glutamine, which is, like I said, obviously the trinucleotide repeat, and it has to occur more than 35 times in order for the Huntington protein to occur. So glutamine is actually an A amino acid that is used for the biosynthesis of the Huntington protein, and it is a neutral charge polar amino acid. So some structural features are that the misfolding is due to that glutamine repeat, and this is shown in the polyQ domain when it becomes strongly immobilized using aggregation. This indicated the significant quaternary or tertiary packing, shown to the right. The addition or interaction with its partner HAP40 is what makes the protein more stable. So moving on to the conclusion and kind of what the current research looks like, such as therapies and etc. So current therapy available is that there's really no treatments that can alter the course and symptoms of the Huntington's disease, sadly. Physical therapy is sometimes used to maintain the patient's current mobility and try and prevent falls from keeping as much current muscle strength as possible. In the future, they foresee stem cell therapy. It's found to be a possibility in which it would be used to replace those lost neurons that are due to the disease. So, current research, I looked up the National Institute of Neurological Disorders and Strokes, the NINDS, and they are currently researching whether additional changes to that HTT mutant gene during development or even adulthood can impact the onset or severity of the disease. They're also researching if the gene could affect the brain's ability to protect and maintain those undamaged DNA, or if that's something that will continue on. They're working to find more biomarkers for the disease so that they can predict and diagnose the disease more easily and quickly and see if that makes a difference. And treatment ways and imaging technology to further their knowledge on Huntington's disease, including their work with skin cells, where they return to the pluripotent state, which is the state in which they can produce more cells, test new potential drugs, and gain more understanding on the neuron malfunction with the Huntington's disease. So I added my sources to the end, and I wanted to also say thank you. I was really interested in researching the Huntington's disease because I am very passionate about mental health. And I feel like a lot of diseases are more so based on the physical component. And this one really interests me because it's kind of both. So I feel like it's gotten a lot of more attention due to its physical, but also one of the main things about it is that it is your cognitive ability. And I think that was really interesting, and I didn't know a lot about Huntington's disease, and I feel like it affects a lot of people, like the facts that were mentioned previously in the United States. And I've learned a lot about it, and I've got to pull in a lot of information from about 319. So thank you for the opportunity, and I've really enjoyed this project. So thank you so much.